So before I started my own group, my work was focusing on the optical approaches to achieve super resolution microscopy. And since ever, I started to do more combination of the chemical approaches and the optical approaches. The chemical approach thrived basically from the second year of the Nobel Prize in 2014, which was given to the optical approaches to super resolution microscopy. So 2015 is the kind of the benchmark year for a new approach started to be popular in the field. That is the expansion microscopy. I'll go over the two approaches with my talk and focusing on the label retention expansion microscopy, which my lab developed, and then its applications in multiple biosystems. First, I want to basically share with you the different scales now we're looking at when we talk about super resolution microscopy. And these different scales is defined by biologists. For example, cellular scale, you can easily separate cells and see the boundaries of cells and to track the cells with conventional fluorescence microscopy. Its resolution is limited by the diffraction limit, which is approximately half of the wavelength. So for example, if you're using 500 nanometer excitation light, and then the diffraction limit of that conventional fluorescence microscope will give you is about 250 nanometer. So that's where that number comes from. I believe all of the audience knows the diffraction limit. And if you want to see more details in the cells, for example, how subcellular structures, for example, organelles or protein complexes were organized spatially, or track their life activity, the organelle-organelle interaction, or single molecule organelle interaction. Then we go to the super resolution microscopy regime, which covers from 10 nanometer to about 100 nanometers. So anything below, well, if the microscope could achieve a resolution below the diffraction limit, it's called super resolution microscopy. But if you want to go further to see for example, to track single proteins, single molecules in the live cells or fixed cells, you go to the electron microscopy field, which gives you 0.2 to 30 nanometer resolution. And now, of course, electron microscopy currently primarily only handle the dead samples, the fixed samples. So if your biological systems are, or your biological questions are at different scales, the ideal way is to choose the resolution or method matching that scale. It's not true that the higher the resolution is, the better the approach is. So whatever matching would be the most ideal method you should choose. So coming back to the super resolution approaches. So we started from the optical approaches since 90s in last century. One of the most well-known approach is single molecule localization microscopy, including the Palm, Storm, STAD, and many other methods. And there are other optical approaches, for example, structured light or detection. For example, SIM is the widely used method, structured illumination microscopy. For this regime, it gives you slightly worse resolution, which is about half of the diffraction limits. But the speed of the imaging is faster than the single molecule localization microscopy. For single molecule localization microscopy, for example, Storm or Palm, you basically only, you have to spend about 10 minutes to get a reconstructed super resolution image at the resolution of around 15 nanometer. So there is always a trade-off for the super resolution. And there are other methods, for example, fluctuation imaging methods. They give you about 50 nanometer resolution. There are many more that I didn't cover in this slide. But they basically design a different optical arrangement in the microscope to bypass the diffraction limit. And starting from 2015, a new approach started to get attention in the super resolution field. The benchmark method is called expansion microscopy. It's Ed Boyden Lab at MIT or Broad Institute initiated this method, expansion microscopy. From the name, it sounds like a microscopy method. However, it's just a chemical sample preparation method. It doesn't require specific microscopes at all. So instead, it enlarges the sample itself. Imagine you use the same microscope, but having a larger sample, basically isotropically in XYZ by 3 to 10 times each dimension. And then you effectively increased the relative resolution of your sample. So you increase the resolution of your existing microscope by the factor of expansion, the length factor, the length expansion factor. I'll explain how we achieve that. So first, this method embeds the cells or tissues into a swallowable hydrogel. Sometimes I make a joke for this method and calling it the diaper science. The reason is the material that we use to embed the cells and the tissue is very similar to the formula used in the diaper. So it's a swallowable polyacrylamide hydrogel. And we cross-link the proteins, RNA and DNA into the hydrogel scaffolding. And after that, by adding water, you can expand the sample together with the gel by typically 4 times and maximally 20 sometimes. So you ended up with a much larger sample on your microscope, and you can see more details with the same diffraction limited resolution. But it's non-trivial to achieve uniform expansion. We definitely want to keep the relative spatial relationship of proteins or organelles after expansion. Otherwise, we lose the point if we introduce distortions or artifacts into the structures. To make sure that the expansion is isotropic in the cell, we have these several steps. First, you stain the sample with your normal immunostaining routine. It could also be protein tags like SNAP tag, clip tag, and then you decorate your cells, all the proteins, with a acrylamide handle. And then you form the gel in situ, meaning that you embed your sample into the liquid monomer of the to form the polymer, and the monomer can penetrate the cells and get into every corner inside of the cells and inside of tissues, and also outside of the samples, and form a hydrogel inside out. So that's so-called in situ hydrogel. Theoretically, most of your proteins, if you are interested in RNA and DNA, you can also cross-link the RNA DNA onto the hydrogel. And then there is a critical step, proteinase K digestion. So after the gel is formed, you incubate the gel with the cell inside of the gel into the proteinase K buffer and do overnight room temperature digestion. The point of this digestion is to break down the protein-protein interactions inside of the cell. So when the time comes for expansion, the neighboring proteins don't hinge each other, because after the protein is K digestion, the interactions between the neighboring proteins break down, and these proteins are free for expansion together with the hydrogel they covalently cross-link to. Okay, after the digestion, or you can call it the protein denaturation, because you can also use heat to break down the interaction of the neighboring proteins. And then after that step, you just add the pure water into your hydrogel, and your cell will be expanded isotropically together with the polymer. Okay, per experiment, this whole procedure only costs you a few dollars, and then you end up with a much larger sample, which is typically 4 times larger in each dimension, X, Y, Z. And the good thing about expansion microscopy is not only it's cheaper, but also you can use any existing system to achieve existing non-super resolution system to achieve super resolution with this larger sample. And that gives you another benefit, that is you can achieve higher imaging depth in this way. For example, all these optical approach super resolution microscopy methods listed here, most of them have a limitation for the imaging depth. For example, STORM can only guarantee you the super resolution for 2 microns in Z. Beyond 2 microns, you lose the super resolution. Similarly, SIM is also not that good with the imaging depth. And if it's more than 2 microns, again, you lose resolution. But for expansion microscopy, since you now are free to degrade, to downgrade basically the microscope needed for the similar super resolution, you can go for confocal to achieve 70 nanometer resolution, and your imaging depth is defined by the confocal. Okay, and the combination between the expansion microscopy is not only limited to the confocal. It can be combined to any optical microscopes. And the goal of my lab is to combine the optical approach and the chemical approach together to achieve molecular resolution imaging. And the third branch is to use machine learning to further improve the images taken with this combined approach. For example, denoising or machine learning based super resolution to push forward the resolution even more. With that, the combination is easy or straightforward, because it's basically just expand your sample and put your sample on top of any microscope. And if that is successful, you basically can push forward the resolution of optical microscopy to the molecular resolution regime, and push forward to the conventional microscope resolution to the super resolution regime. Then it would be straightforward to any biological research labs to achieve it would be easier for them to achieve molecular resolution without a electron microscope nearby. The access to good electron microscopes is the major limit for the researchers here to get molecular resolution images. And I think it's the same for every country. So I think using pure optical systems to reach to the molecular detail will help the research a lot. And if that comes true, so this thing is straightforward. However, when we start to combine the optical approaches, the super resolution optical approaches with the expansion microscopy, problem happens. We found there is a severe challenge in expansion microscopy. That is very serious fluorescence loss after all these procedures. So the samples labeled with organic dyes lose about 50% labeling after the whole expansion procedure. And this fluorescence loss is basically the quenching or loss of label, it's not because you expanded the sample and the signal is diluted. It's not because of that. So 50% loss is the real loss of your label. And the worst thing is the best photo switchable dye, which is used in Storm or Palm, and sometimes instead is suffering the most fluorescence loss by about 90% loss. That means after the whole procedures of expansion, digestion, you end up with no signal. So we were pretty disappointed by this. We didn't expect this will be a very serious troubleshooting or an independent long project. So to conquer this problem, we need to figure out what's the causes to the fluorescence loss in expansion microscopy. Basically, 2 steps in the procedure cause the major fluorescence label loss. The first one is a polymerization step. So the polymerization step is a free radical chain reaction step. So there are a lot of free radicals and electrons going on in the system around the cell, around the labels inside of the cell. So these free radicals can react with the organic dye to change their conformations and structures. So they are in the dark state permanently, and they will not fluoresce anymore. So this caused about 20% of the loss. And the other unexpected major cause is the proteinase K digestion step. Since the digestion is so the proteinase is working on chopping the proteins. So sometimes when it's overdone, it can make one protein fragmented. And as you can see, the organic dye, which is these green dots, is cross-linked to the polymer, which is the black wiggles here, through the antibody, which is the yellow thing, because only the antibody or other proteins are decorated with that acrylamide handle. So if we proteinase K chop or fragmentate the antibody, then if the dye molecule is not binded on, is not conjugated on the fragment, which happens to be chemically cross-linked to the hydrogel, then you permanently lose that probe by washing them out during this expansion step. And this contributes to about 30 to 40% of label loss. To solve this problem, we started with this chemistry, a new chemistry label. We call it trifunctional label. And the goal of this method is to retain those labels into the hydrogel for the downstream imaging. So we call this method label retention expansion microscopy, short as LR-XM. The key is a series of trifunctional labels. We don't use organic dye at the beginning to immunostain the samples anymore. Instead, we immunostain or we tag the target proteins with trifunctional labels. Each of the label has 3 functions. The first one is the reporter. So a reporter is a small molecule biotin or destruction gene or azide. So these small molecules can survive the polymerization, which means the free radicals don't quench them or change their structure. And another thing is these small molecules can be labeled with fluorophores after expansion. So after all the label or organic dye damaging steps, you can introduce safely the fluorophores through these molecules binding partners, for example, fluorescently labeled streptavidin that combines to the biotin in your trifunctional linker to introduce the fluorophores at the very last step before your image. so your fluorophores don't meet any harsh chemicals anymore. And the second functional group is the connector. For example, NHS ester that could bind to antibodies or that could bind to antibodies, and your antibody will bring this trifunctional linker to the protein of interest. So instead of conjugating the antibody with the dye, we conjugate the antibody with this trifunctional linker. And the connector is the chemical group that will covalently bind onto the antibody. And the connector can also be some ligand to recognize protein tags, for example, snap tag ligand or clip tag ligand or halo tag ligand. If you have these tags fused to your target protein, you can just throw in these trifunctional linkers with those ligands specific ligands. And the third functional group is the anchor group, which is the methacrylamide in our case. It functions similar as the acrylamide. It's just stabler than the acrylamide. Methacrylamides will covalently react with the monomer while the gel or the polymer is formed. So its function is to covalently cross-link the probes onto the polymer. So during the digestion step, even if you lose all the proteins, you can still have the label in place in the original place, because this trifunctional linker just covalently cross-link the reporter onto the polymer. So with this trifunctional linker, we basically could solve the eliminate the probe loss during the polymerization and the digestion steps. And this work started several years ago when I was still a postdoc at Bohuang Lab, and I started collaborating with Ian Staple Lab. Chi and Arthur are Ian's students and postdocs. And Ian is a chemist professor at UCSF back to that time. And then I moved over, and Andrew McCulloch is my postdoc, was my first postdoc, and he carried on this project. And we found new chemist collaborators at UCI from Vidal Lab, and Alex and Kirsten are Vidal student. And I also have this chemist collaborator, Zhipeng Dai, who followed the whole protocol from the very beginning to now. So these are the novel trifunctional labels that we use. These are different labels to introduce multiple colors or to bind onto antibodies or ligands differently. And using these trifunctional linkers, we modified the procedure of the expansion microscopy to the LR expansion microscopy protocol. The biggest difference between the LR expansion and LR XM and the standard XM are two steps. That is the first step and the last step. The first step, instead of adding in the organic dye conjugated onto antibodies, we are using the antibodies conjugated with our trifunctional linker. That's it. Or using the trifunctional linker targeting the protein tags, for example, halo tag, snap tag, directly. So the middle steps are identical to the standard expansion microscopy. If you already know how to do that, those steps are the same. So gelation to form the gel, digestion to break down the protein-protein interactions, and expansion to make your sample larger. And then we have this additional step, post-expansion labeling, to make the sample fluorescent. So here is when we add in the fluorescently labeled streptavidin or fluorescently labeled anti-digoxin antibody for two-color imaging. And now it is time to check our result. So the system we use to calibrate to validate our result is clathrin-coated pits in U2S cells. For the size of clathrin-coated pits is from 100 nanometers to a couple hundred nanometers. So with conventional microscopes, you cannot see the hollow structures of the clathrin-coated pits. Each of the dots that you are seeing is one clathrin-coated pit. And with expansion microscopy, this is a standard expansion microscopy with the Alexa fluor 488 dye, and you can see the samples are enlarged. And in these structures, you can almost see the hollow structure of expanded clathrin-coated pits like this one and that one. So you may see a little bit dimmer in the middle. However, because the signal-to-noise ratio is not that great due to the fluorescence loss, you cannot see them confidently or clearly. But with the label retention expansion microscopy using the trifunctional linkers, you can clearly see the structure, the hollow structures of clathrin-coated pits like this and that. Because the signal-to-noise ratio is higher. So the LR expansion, in this case, the LR XM is 5-fold brighter than the standard XM because of the retention of those fluorescence labels. And now we're ready to combine the expansion microscopy with the STORM because we solved the fluorescence loss problem. And again, we use clathrin-coated pits as our model system to validate of our resolution, our molecular resolution. The reason is if you use electron microscopy to image the clathrin-coated pits, you will see they are not only a hollow structure, but also a hollow structure formed by hexagons and pentagons. Because the distance between the vertices on the clathrin-coated pits is is less than 20 nanometers. And you do need less than 10 nanometer resolution to be able to separate the neighboring vertices. And in our case, we labeled all the vertices with a snap tag. So the green, the purple shows you the structure you expect to see when you focus on different Z on the clathrin-coated pits. For example, if you focus on the top of the clathrin-coated pits, you should see all the polygons. And if you focus on the middle, in the middle of middle cross section of the clathrin-coated pits, you see a hollow ring, right? Because it's a hollow structure. And this is in the middle is how we label our clathrin-coated pits. This is in cell, it's in situ. So we just use the CRISPR-Knock-in gene editing to introduce the probes into every clathrin. The probes are the snap tag. I think in this one we used overexpression, not the knock-in. And if everything's ideal, we expect to see separated clathrin labels as vertices at different levels to give you different imaging. And first of all, let's see if it's necessary to use expansion, to combine expansion with STORM. So first we image the labeled, snap tag labeled clathrin-coated pits in the cells with STORM, and we don't see the structure we expected to see, which are the separated dots to form polygons, because the resolution is not there yet. And then we combine the STORM with LR expansion microscopy and achieved 4.5 nanometer resolution. Here, the resolution is the scaled back resolution. The resolution for the STORM microscope is still like about 15 to 20 nanometers. The 4.5 is that resolution divided by the expansion factor. And here, only with 4 times expansion, you can see the separated labels, snap tags on the at the vertices of those polygons. And in the when you focus the objective through the middle cross-section of the cathode pits, you see the discontinuous ring as what we predicted or what we designed in the labeling fashion. So we're happy with the result, but you're also seeing a little bit distortion, right? Because ideally, at least they should look like the distributions of the vertices in the like in the electron microscopy graph. However, you don't see the organized with equal space. The major reason is the size of the tags of the tag we label our system. So this is one new thing we need to pay more attention to. So when the resolution is at around 20 nanometer-ish, you probably don't need to think too much about the label size, the size of your label. For example, if you use SnapTag, which is smaller than 3 nanometers or 5 nanometers, you don't need to worry too much about the size. You probably need to worry about antibody, because the size of antibody is about 15 nanometers. That's close to the regular super resolution. For example, 15 nanometers. And now, like, because we moved to the molecular resolution, which is around 5 nanometer resolution, then a size of a label around 2 to 3 nanometers will cause some uncertainty of your localization of the target protein. So that's one thing we're working on. But I think that's a sweet problem to troubleshoot. And now let me summarize the method itself, and then we'll move to the applications. So how to calculate the effective resolution of expansion microscopy? First off, the equation is the effective resolution equals to the microscope resolution divided by the length expansion factor. Okay, for example, if you have a conventional fluorescence microscope that gives you 250 nanometer resolution, and you expand your sample 10 times, like what we are doing in this slide, then your effective resolution will be 250 nanometers divided by 10 times expansion factor. That will give you about 25 nanometer resolution, effective resolution on your conventional microscope. Okay, so this image shows you before expansion, the gel. This is exactly the same gel. So before the expansion, your gel with the cells inside of them, they're very transparent. You won't see the cells with the iPhone taken image. So before expansion, there are very small, like about 5 millimeters. And after expansion, on the that is the image on the right, you can see the gel can really absorb water and grow really big in 3 dimensions. And here what we achieved is 9.3 times expansion. So for your experiments, you need to measure the after expansion gel size to for every experiment to have that length expansion factor and have your accurate size, so that you can scale back to the original size of your cells and tissues, and accurately calculate your effective resolution. And the gel, basically, with different formula of the gel, you can control the expansion factor. Some formula of the monomer can give you a really, really large gel, and we have achieved like about 13 times expansion, like 1 round expansion with some gel formula. But the typical one we're using is 4 times, because that already gives us enough resolution for most of our studies. And also, even with the same gel formula, you can also let the gel stop at a certain stop expansion at a certain length expansion factor by control the expansion buffer. Now, if you use pure water, you expand your gel maximally. That is defined by the gel formula. But if you use slightly salted buffer, you can achieve but you will shrink the gel a little bit and have a smaller gel. It really depends on what you need eventually. Now let's move to the application of the LR expansion microscopy. The first demonstration is in the cell, in the organelle called the nuclear lamina, which is a meshwork underneath the nuclear membrane. And there are meshwork reviewed by the electron microscopy decades ago. However, the electron microscopy cannot tell us the specificity of what lemon proteins or what lemon fibers are forming these meshwork, and how different lemon proteins form the structures together. Lemon structure is very important for human health because there are very associated with our aging. So if mutations happen in lemon A gene, then so there are 500 different mutations which could happen to the single gene and cause progeria, meaning that a person will age much faster than normal. And also in our body, like even if we don't have those mutations, our nuclear lamina structure will change when we age. So it's very important to study its structure and function. So this is a project as a collaboration between my lab and Enrico's lab. So Lorenzo from Enrico's lab is helping us at the first stage of this for the imaging for this project. And my student Yin is now leading this project, and Andrew, the postdoc, helped her at the beginning. So without expansion, this is what you will see with the epifluorescence microscope for the lamina. They look like a hollow structure, hollow sphere around the nucleus. With the LR XM plus confocal, you can see beautiful meshwork formed by this is lamin A, and also you can see the you can have the 3D structure straightforwardly, and this is almost like as far as I know, this is the first time we use the optical microscope to see the meshwork without overexpression of the lemon protein, and that agreed agreed very well with the EM image when we moved to the area scan. So the LR XM plus area scan will give us about 30 nanometer resolution, and we started to see the finest meshworks that could register with the SEM image. And also you can have multiple colors in the image to the organelle interaction. So here, the cyan color is the lamina meshwork, and the red color is the nuclear pore complexes labeled with anti-NUP153 antibody. So if you look closely, you will see this anti-localization between these two organelles. Anti-localization here means the nuclear pore complexes seeds into the holes of the lamina meshwork. And here is just a demonstration. We already know the reason why they avoid each other, because lamina is connecting with the heterochromatin to suppress the gene expression, and the nuclear pore complexes connects it to the actively transcribed genes. So they have opposite functions, and that's why that makes sense. They avoid each other in space as well. But this is a demonstration with a known function. There are so many interesting spatial relationship we're seeing, like new spatial relationships, and we currently don't know their functions, and we're studying towards that direction. And the LRXM, of course, can be used in tissue imaging as well. So this is a mouse brain section. And with conventional method, conventional microscope, 10x objective, this is the astrocytes you can see. And with LR expansion microscopy, same microscope, same objective, you can see much more details of the GFAP distribution in astrocytes. It's a cell type in brain, an important cell type for Alzheimer's disease and many other diseases. And with the same method, but different objective, you start to see a lot more detailed structures in the astrocytes. And last part will be our turnkey AI-driven super resolution microscopy. It's more a imaging processing method. So basically, we use synthetic training set to train our AI network and to make them recognize our structures. And to save time, I think I'm running out of time. I'll just show you the results. So from the input low-res image, we can achieve very high super resolution AI output. Comparing with our ground truth, it's pretty close to what the reality is. But we are still working on this AI-driven analysis. The best thing is that you don't need to train this network. It's trained already, and it could apply to images taken with different modalities, meaning taken in any lab without additional training. And also this AI can enrich the network that we just imaged or any other structure like that, and help us to quantify the features of the structures, for example, the whole area in the network or the fiber density in the laminar meshwork. To summarize, the advantages of combining the optical, chemical, and computational approaches, for example, LR expansion, microscopy with STORM, we can achieve 5 to 70 nanometer super to molecular resolution, depends on what microscope you put your enlarged sample on. And we'll have bright signal, low background, because the gel lesion itself is the gelation and digestion itself is a sample clarity method. So that's why you also suppress the background. And it will give you high imaging depth. If you use 2 photon, it could go up to 1 millimeter or multi-photon. And also we can have quantitative imaging with that. And the applications are, now we are really moving to the molecular resolution. So a lot of things super resolution microscopy cannot achieve now can be seen. For example, the protein-protein interactions. But of course, it really depends on your what microscope you use and how large you expand your cell. It can definitely handle a wide range of super resolution imaging of protein complexes, RNA and chromatin, organelle structure, organelle contacts, and also thick tissue, because it's cleared sample during this whole expansion procedure.